In this presentation, I will introduce mitosis in the basic usage of the microscope, which was the lab session I did in Biology 110 at Duke Queensland University. And the main focus will be on mitosis. I'll first introduce the background information based on what I've learned about mitosis in this course. Then I'll explain the steps in this lab experiment in detail. Finally, I'll discuss how mitosis is related to the real world. Let's start with what is mitosis. Mitosis is a kind of cell division that results in two daughter cells with identical genetic information. The cell cycle usually consists of mitotic phase, which is known as the end phase and contains mitosis and cytokinesis. The interphase is consisted of the cell cycle as well, which takes up a large proportion in the cell cycle, but the exact percentage varies from cell to cell. Now let's take a closer look at the interphase and the end phase. The interphase can be divided into three parts: G1 phase, S phase, and G2 phase as the abbreviated names of the first gap, synthesis in the second gap phase. In G1 phase, the cell mainly synthesizes proteins related to RNA and cell division. Then comes the S phase, where the chromosome duplicates. In the G2 phase, spindle microtubules assemble and duplicate centrosomes in animal cells. What's more, there are three checkpoints during the cycle to form a cell cycle control system, which gives out go-ahead signal at each checkpoint to make sure the process of whole mitosis. When the interphase is passed, the mitosis starts. The P for prophase, the N is for metaphase, and the A is for anaphase, the T is for tagphase. So remember P and P, the stage in order. The very first step is prophase. Prophase because it's the beginning step, the nuclei is still there, and it's going to go away later on, but this is the stage where it's actually still there. The chromosomes are visible. In fact, we'd say they're descending, which means they're thickening and visible. The next stage is metaphase. N for metaphase, but I also like to remember N for middle, because in this stage, the chromosomes line up in the middle of the cell. The nucleus, we've got chromosomes waiting there. Next, the A is for anaphase. Anaphase, I like to think of the A for away. So the chromosomes move away, they're moving to opposite sides of the cells, so they're moving towards the poles of the cells. Now, one thing to point out, these chromosomes, they're not moving by themselves, they actually have something called spindles. The spindles are fibers that help move the chromosomes to the ends, kind of help them move along. And the last stage is mitosis. Think T is for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and nuclei are forming on each side to make these two new cells. Nuclei is starting to surround the chromosomes on both sides. I like to think the T is for two because you can actually see in the step that the end goal is going to be two cells in the human body. They're going to have 46 chromosomes. And again, remember they are identical. Cytokinesis is responsible for the final separation into two cells by splitting the cytoplasm, which completes after the PMA mitosis stages. So why did all of them matter? Without understanding cell division, we wouldn't understand how growth and repair happens because they both require more cells to be. Next, I would like to introduce how to make the specimen used in the growing root tips of garlic. After the specimen is made, we use microscope to observe the mitosis. The purpose of this lab are as follows. First, learn how to use microscopes. Second, learn how to make garlic root tip slides for microscopic observation. And third, observe cells at different stages of mitosis. In this experiment, we will use the following materials and the equipment. First, cut a few roots better to be 1 centimeter from actively growing garlic root tips using a seeder and place them into a glass container filled with fixative solution. In this case, the fixative solution is a mixture of ethanol and acetone at the ratio of 3 to 1. Make sure you pour enough fixative solution to cover the root tips. Leave the root tips on the sternum for 2 minutes. The purpose of this step is to dehydrate the cells to fix the chromosomes in the cells. Then we transfer the root tips to a glass watch and cover the root tips in a few drops of 1 more hydrochloric acid for 5 minutes. The purpose of this step is to soften the cell walls so that the stain can easily penetrate the cells and stain the chromosomes. You should avoid contact hydrochloric acid with your hands. Now use 4 steps to transfer the root tips to another clean watch bath. Add 2 to 3 drops of the 0.8% intense violet stain of the tips and then allow the root tip to soak in the stain for 3 minutes. Use paper towel to blot away excess stains. Add a few drops of distilled water to the stained root tips. Use paper towel to blot away excess stains. And water. Repeat the washing process 1 more time and use paper towel to blot away excess water. Add 1 drop of distilled water to clean gas slides. You can simultaneously prepare a few slides. Use 4 steps to transfer 1 root tip or 1 of the gas slides in the drop of water. Cover the tip with a cover slip to avoid generation of air bubbles. Make sure 1 edge of the cover slip touches the gas slide, and then gently lower down the cover slip. Use the razor end of the pencil to apply pressure on the cover slip to squash the raw tissue. Make sure you'll be gentle not to break the cover slip. Make sure you're gentle not to break the cover slip. The slide is now ready for microscopic observation. The propelled garlic root tip slide under microscope and at high and low power using microscope. Try to find cells at different stage of mitosis. You should be able to see chromosomes at different stages of mitosis as shown in the picture. Then I want to discuss the relationship between cancer and mitosis in the real world. What is cancer? Your body is made up of millions and millions of cells. In fact, there are between 50 and 75 trillion cells in the body. These cells are dying and being replaced all the time. Your red blood cells live about 4 months, white blood cells on average for over a year, whereas your clone cells have a lifespan of only about 4 days, while your brain cells last for a lifetime. Cancer can start when just one of the trillion cells begins to grow and multiply too much. The result is a mass of cells called a tumor. The starting place of abnormal cell growth and division is called the primary tumor. They can start almost anywhere in the body changes take place. Within the genes of a cell, the group of cells resulting in abnormal cell division genes are specific codes of DNA that tell the cell what to do by coding for specific proteins or RNA molecule proteins. In order to get control the cells that decide what type of cell it is, what it does when it divides and when the cell will die. When cells divide by mitosis, the DNA is replicated and sometimes mistakes are made. These are called mutations. Lots of mutations are silent, they have no effect on the cell because they occur in non-coding regions of the DNA. But sometimes mutations in certain genes may mean that too many proteins are produced that trigger the cell to divide, or proteins that normally tell the cell to stop dividing may not be produced, so then start to grow out of control. But don't panic, this isn't quite as risky as it sounds. There has to be roughly 6 or more mutations to coding DNA before a normal cell turns into a cancer cell. So mutations do the damage, but what caused them to happen? Cells are usually very good at repelling damage to genes. They have special repel mechanisms in place, but over time the damage may build up. And then it can be a domino effect. Once the cell starts growing too fast and dividing rapidly, they're more likely to pick up even more mutations and thus likely to repel the damage. While these mutations can happen by natural processes, they can also be triggered by lifestyle. For example, there are cancer-causing substances known as carcinogens in tobacco smoke. In fact, more than 4 out of 5 cases of lung cancers are caused by smoking. Genetics also have a role to play. 40 genes can be passed from parents to offspring. Most inherited cases of breast cancer are associated with 2 abnormal genes. BRCA1 and BRCA2, for example. Another cancer-causing factors include exposure to radiation exposure to UV radiation from the sun, and some chemicals in the environment, among these other things. So, yeah, from this video, you should know what cancer is, the result of changes in cells that lead to uncontrolled cell growth and division. It is a non-communicable disease in humans, which means it is not infectious cancer. It is caused by changes in a person's DNA, and these changes cause the cell to divide by mitosis more than it usually would. Yeah, so this is almost the end of this presentation. I want to say thank you to Professor Lee, who is so kind and patient to all of us. I want to thank everyone in BIOL110 who gives me lots of support throughout this session. Finally, I want to thank my university DKU for offering me great lab experiences. Thank you so much for watching.